Good afternoon, everyone. Welcome to the Lunchtime Lecture series from BRIT and the Fort Worth Botanic Garden. I'm Dr. Best, Director of Research Programs, and I and my research colleagues host this lecture series, which takes place the first Tuesday of each month. Our next talk will be May 4th, when Dr. Rebecca Cole from the Perot Museum will present Permian, Texas, Exploring an Ancient Tropical Landscape. We are streaming live the lecture to YouTube, and you live viewers, you can type your questions there anytime, and we will answer them at the end of the lecture during the Q&A when your questions will be displayed on screen. So you might want to keep them kind of short. If you'd like to test out the chat now, though, feel free to share where in the world you're watching from today. I want to mention that this lecture and the accompanying live chat are being recorded and will be available in the future on BRIT's YouTube channel. And if needed, links to other resources mentioned during the talk will be posted on the BRIT webpage for this event. And if something happens and we get cut off, please refresh your YouTube page or keep checking the event page at brit.org, as we'll be posting new information in both those spots about possibly reconnecting or switching to a new link or even possibly rescheduling. So let's get started. It's my pleasure today to introduce today's speaker, Dr. Peter Fritsch. Dr. Fritsch holds two titles here at BRIT, Vice President of Research and Director of the Herbarium. He received his PhD from Rancho Santa Ana Botanic Garden and Claremont University, and was a postdoc at Duke University after that. Before coming to BRIT in 2015, Peter was a curator of botany at the California Academy of Sciences. And I have to say that, boy, we really lucked out when Peter came to BRIT. Not only is he a great scientist, but he is a phenomenal grant writer. In under 6 years that he's been here, Peter's been PI on 4 major NSF awards and co-PI and a collaborating institution on a few others. So that's just really saying something if you're familiar with the process. Today, I believe he'll be talking about a bit about his blueberry work, for which he was just awarded one of those big new NSF grants late last year. So with that, take it away, Peter.
Well, great. Thanks for the introduction, Brooke. I appreciate it. Sound coming through okay? Just want to make sure. Okay, great. All right, so today I want to talk to you all about work that I and colleagues have been doing in the blueberry group, so evolutionary origins and species diversity in the wild blueberries, tribe Vaccinieae. So for much of my research, I focused on families related to the blueberry family, namely storaxes and sweet leaves. Relatively obscure, moved into Ericaceae around 2006 when we started to study the wintergreen genus, that is Gaultheria, and eventually moved in to its kind of sister group, which is Vaccinium or blueberries. And I'm finding that there's multiple careers worth of taxonomic evolutionary questions to address in this group. It's just overwhelming, but yet exciting. And what we really need is an Ericaceae working group with many colleagues collaborating together, as they've done for other large families such as the melastomes. I think that's really a needed step. Okay, so who doesn't love blueberries? I mean, they're a superfruit, right? They're high in phytonutrients and dietary fiber. They taste great. They're one of the few major food groups indigenous to North America, and they're one of the newest of cultivated crops. The first cultivars were planted out in 1912 in, of all places, the bogs of New Jersey. Yes, New Jersey has bogs. And they're the star of a multi-billion-dollar ag industry with festivals, and everybody's got their favorite blueberry patches where these things grow wild, and you can make obviously all sorts of foods and snacks out of them. There's even a beer made with blueberries. So there you have it.
Hey, Peter.
Yeah.
I'm sorry to interrupt. So this is we're having a small technical difficulty. It looks like we're actually streaming on the YouTube site for the next lecture for the May talk. I apologize. That's 100% my goof. Yeah, so just give me one quick sec. I'm going to go over to your YouTube page and change the information so people will come see us on the other channel. Just hold tight. If you'd like to just riff on blueberries in the meantime, go ahead. Oh, This is,
Yeah, there's always some with the technology.
yeah, but that's okay.
Do I need to do anything on my end?
No, you're fine.
Okay.
You did everything. You
Okay.
did everything perfect.
Oh.
I'm just going to go drop that information there. Let me find it real quick. It's interesting. I have everybody texting me being like, where is it? It's not happening.
Okay.
So let me just fix it real quick. It
I
shouldn't
can start again too.
Oh no, it'll be there. People will be able to go backwards. It'll just
All
have the
right.
wrong name on it, and I'll go
Okay.
back and fix that.
All right. Mhm.
Yeah, it says we're live. Okay. Let me see. One moment. It was going too smoothly. I knew it. Okay. Have to grab the other link. Okay, here's this YouTube link. Copy link. I like to mess up on purpose every once in a while. It makes me feel human. Please, link has changed. Let's see. Okay. I think that should handle it. Let's see, refresh. Okay, let's. I'll let you go ahead and just pick up where you left off. That should be good.
Good. Okay. All set, right? Can you hear me okay?
Yes, Peter. Yeah.
Okay,
go
great.
ahead.
All right. So the blueberry slide, superfood tastes great. There's even a beer made out of it. I might have one or two of those beers after I'm done with this talk. And there we have it. So, all right. So maybe the only person who doesn't like blueberries in the world is poor Violet Beauregard in the Willy Wonka movie. Perhaps you'll recall Violet chewed the gum she wasn't supposed to chew because it wasn't ready yet, and she turned into this blueberry. Now, in the process of turning into this blueberry, she may have been contemplating, possibly, really, what are blueberries anyway? What are their specific and generic limits? Are they monophyletic? Does the presence of polyploidy affect estimates of species diversity? Has ploidy change arisen primarily through autopolyploidy or allopolyploidy? Is hybridization truly rampant, or has this been overstated? Well, hopefully she'll have some of the answers by the end of this talk today, but a lot of those questions are still lingering, and it's just going to require a lot of research to solve. So with that, I'll overview the talk, and so I'll first focus on the tribe Vaccinieae, and then zoom into the true blueberries, Vaccinium section Cyanococcus. And then at the end, since this is part of a We Dig Bio event extending over 4 days beginning yesterday, I'll talk a little bit about the benefits and limitations of specimen data mobilization in the context of the blueberry research that we're doing. Okay, so first of all, Ericaceae is the heath family. Maybe many people know with this, just familiar with this family from rhododendrons, blueberries, et cetera, et cetera. But it's a huge family. It's one of the largest families in the angiosperms. And it's part of this order Ericaceae, which is 24 families, which included things like kiwis and persimmons and all sorts of primulas and all sorts of cool plants. Well, anyway, the Ericaceae, they seem to like acidic soils. That's pretty, not ubiquitous, but definitely a general trend. They also have these, maybe because of that, they have these specialized mycorrhizae. They're called ericoid mycorrhizae. And they're also mainly woody. At least some part of the plant, sometimes even if it's just above, below ground, is woody. They generally have 5 fused sepals, 5 fused petals, 10 stamens, 5 united carpels. Strangely, they have these inverted anthers. So the anthers invert during development, and so the top is actually the technical bottom. And those anthers usually have pores, and sometimes they have slits. And the pollen's kind of cool too, because the pollen is in these tetrahedral tetrads, which is quite distinctive. So within that family, the heath family, we've got the tribe Vaccinieae, a large tribe of 46 genera, over 1,600 species. And it includes, say, not only blueberries, but also related things like cranberries. For example, the bottom red fruit there is a cranberry. The one on the left, far left, is a bilberry. The bilberry doesn't have a crown like most blueberries, and so you can always tell a blueberry from a bilberry from a blueberry that way. And there's also huckleberries in this group. So let's see, I'm going to expand this a little bit here. And so the inferior ovary is the way to tell Vaccinieae from any other Ericaceae. There's no other group that has it. And so, and the other way to tell is through these anther tubules. And so other genera, some other genera of Ericaceae have those tubules, but it's most well developed in this tribe. And some other characters are found, but only in combination do they really indicate Vaccinieae. For example, this abortive bud and the vegetative part, if you see that abortive stubby part there, then you know you've got Vaccinieae, except there are a couple other genera that do that. But all Vaccinieae do that. And also the berry. All Vaccinieae have a berry. And so other genera of Ericaceae have a berry, not very many. I can think of like the crowberry, for example, and Petrum has a berry, but generally speaking, if you've got a berry, you're in this tribe. Okay. Let's see. Okay, here's some other fun features of the tribe. A lot of them are epiphytic, believe it or not. That means they occur on other plants, big trunks of trees and things like that. The anthers often have these spurs on the back. That's thought to function in pollination. Some have, some don't have. Sometimes there are these large bracts on the inflorescence, and those can be persistent or deciduous. There's these cool extrafloral nectaries on the bottoms of the leaves, and that's almost ubiquitous. Some don't have it, but most of them do. Even the cultivated blueberry has some form of glands down there. And interestingly, too, they're not well known. They're not understood. They're not well documented morphologically. They're not used in keys, even though they vary quite a bit. It's quite interesting. And then there's they can be deciduous, and they can have serrate leaves, or they can be entire, and then they can have these real thick leaves. A lot of the tropical ones have that kind of planar veination you see there on the lower right. There's tremendous variation in Vaccinieae flowers, and so you can have all different colors. The tubes are, corolla tubes can be long, it can be short, multicolored zigzag patterns, candy cane stripes. It's pretty spectacular. The long ones are probably long-tongued insect pollinated and even bird pollinated, whereas the short ones are often buzz pollinated. So that means that the insect vibrates its usually a bee, well, it's a bee, and they vibrate their thoracic muscles and then kind of vibrate the pollen out of those tubules that way to get to the pollen. They're going for the pollen when they do that. Well, so focusing on Vaccinium, the blue line there So everything to the left of the blue line is non-Vaccinium. Everything to the right is in the genus Vaccinium. And so there's a big problem in that Vaccinium really, because it's not, they're flowers that really aren't as specialized as some of the other genera with easily identifiable features. They just have relatively short flowers instead of long tube flowers, and that's kind of been the defining feature. So, for example, in Agapetes, the Florum leysiana key, if it's greater than 1.7 centimeters, then it's Agapetes. If it's less than 1.5 centimeters, then it's Vaccinium. Well, the problem is that there are real short-flowered Agapetes, and of course there are real long-flowered Vaccinium, and there are no other consistent features that separate these things. So this has been recognized in the global treatment of Vaccinium by Stevens et al. In the families of genera of asteroid plants, where they say the genus is wildly paraphyletic and the limits are unclear. And that's reflected in the latest, unfortunately, 2002, so it's been a while, Sanger sequenced ITS phylogeny. That's what we got, as kind of a global, state of the situation in the tribe. So the overall resolution is low in this tree. There's strong support for a whole Andean clade, which comprises a couple of dozen genera. And there's weak support for an old world, new world split. But sampling's not large. Vaccinium's highly para or polyphyletic or not a natural group. And a companion paper to the one I'm referring to indicates that many of the Andean genera are actually not monophyletic at all. So there's a lot of work, really basic research on this that really needs to be done in order to make any headway on what the relationships are in this tribe. Okay, so going to Vaccinium more detail, how many species then are there in Vaccinium? Well, you talk to different people, you get different answers. Flora North America has 500, Flora China, Flora leysiana 450. The families of genera vascular plants 140. They move out a lot of species into other genera. And then Walter Judd, an Ericaceae expert, goes so far as to say, well, there's really only one species in Vaccinium, and it is the type naturally of the genus Vaccinium uliginosum, which is a circumboreal species and named by Linnaeus. And he would put all the rest of Vaccinium, all 30 sections in the genus would go in different genera. So what's right, what's wrong, what's the best thing to do? It's all up in the air still, and it's all waiting to be done. So I have a number of current projects outside of the true blueberries, and I'll talk about the true blueberries coming up. But for now, I'm working with Stephen Smith and his colleagues, and he's got a grant to study the phylogeny of the Ericales group, including the Vaccinieae. But this will not be comprehensive sampling in the Vaccinieae because it's an order-wide phylogenetic analysis. They'll probably include anything they can get their hands on that'll work for sequencing. I'm also working with several colleagues to describe new species in Vaccinium and also in Agapetes in Southeast Asia. So that is, so for example, Kate Armstrong at New York Botanical Garden, Wendy Mustachim in Indonesia, Yuhua Tong in the South China Botanic Garden, Maxim Nuraliev in Moscow State University, and Victor Amoroso at Central Mindanao University. All new species. There's plenty of new species to be described. And as people are investigating areas more, they're finding more and more. I'll also be mentoring with the advisor, John Horner at Texas Christian University, a Filipino master's student, Maverick Tamayo. This is part of another grant that we're working on in the Philippines Biodiversity Survey, and he'll be undertaking a revision of Vaccinium in the Philippines. Finally, I'm on the committee of a PhD student mentored by Nico Selenis. Her name is Anna Becker, and she's working on Hawaiian Vaccinium, and which is really going to be an interesting story. How did Hawaiian Vaccinium get to Hawaii? How did it disperse across the islands, and how many species are there? She's finding so much variation that wasn't understood. It's just astounding. All right, so to the extent that it's difficult to define what the blueberry tribe is and what some of the genera are, I mean, in the blueberry tribe is what I mean to say, it's actually a lot more straightforward to define actually a true blueberry. Well, what is a true blueberry? It's in this section, Cyanococcus, and cyan is blue, coccus is berry. I believe that's Latinized Greek. And so they're all native to North America. There's anywhere between 9 and 25 species. I'll go over the problems there. And it's got a haploid chromosome, a number of n equals 12. All right. There are two defining features of this group. If you see these features, you know you've got a true blueberry. They don't occur anywhere else in the tribe. It's one is this kind of stripy bark, and it's the way that the periderm develops. It has two layers, and the one layer is a superficial layer that kind of erupts here and there in patches, and that's those white diamonds you're seeing there. So if you see a bark like that, then it's going to be a true blueberry. The other way is to look at the end branchlet, and you can see all those little dots on that red branchlet, and those dots are actually stomata, and they're raised, and so the whole stem is kind of what we call verrucose or warty, and so you've got these warty stems. Sometimes not easy to see, but a lot of times it's pretty straightforward to see, especially on dried specimens. You can easily pick them out. All right, so other ways to tell what a true blueberry is, these are not as definitive, but in combination, they'll indicate a true blueberry. The first one is that they've got differential buds. The floral buds are much larger than the vegetative buds. The flowers are always in inflorescences. Some vacciniums have single flowers, but these are also always clustered. This doesn't have a spur on the back of the anther, so that's actually not as common. The berries, you probably know, or maybe you never thought about it, but they all have this persistent calyx. That's the crown of the blueberry. And finally, the ovaries have what we call a pseudotenlocular ovary. That is an ovary that it actually has 5 locules, but they're almost divided to the base by these invaginations, and so it looks like they're tenlocular. And so you can see that in the one on the right photo as well. So just remember, whenever you're biting into a blueberry, you're actually biting into a mature pseudotenlocular ovary. Yeah, now you know. Okay, so there are 2 main players part of our research on blueberries. The first is W.H. Camp. Camp was a curator at the New York Botanical Garden and a strong contributor to what we call the evolutionary synthesis, where this was a synthesis between genetics, evolutionary theory, systematics, paleontology, and other fields of biology to provide a unified theory of biology in the context of evolution. And so he was a biosystematist, and so that, in other words, it was kind of experimental taxonomy. And so they were studying genetics in the context of evolution to improve classification. And he used the data from his studies to improve not only Vaccinium crop breeding, but also used it as a model for his writings on species theory. He was to some extent eclipsed by folks like G.L. Stebbins, other botanists, partly because of his writing style and use of jargon such as mictons and things that nobody really picked up on. And it was really kind of dense, and for that reason, he's not as well known as some of the other players in the modern synthesis. But recently there was a paper on Camp and his contributions. Okay, so at this point I need to mention polyploidy. Polyploidy is simply an increase in the haploid number of chromosomes in a cell from 2n to 3n to 4n, and so on and so on. And so plants can do this. There's 2 types. There's autopolyploidy, which means polyploidy originating within a species. And then there's allopolyploidy, which means that it originates from two parental species. Okay, and it's rare in animals. Fish do this, for example, but not many other animals. But it's common in plants, and there are possible reasons why that might be. But at least half of all plants have been thought to have undergone some sort of ploidy in its history, whether it's ancient or recent. And it turns out that it's important in true blueberry evolution. So what are the mechanisms for polyploidy? There's thought to have been maybe four main ways that polyploidy can occur. Three of them involve what we call unreduced gametes. So that's a gamete where a mistake has been made. And so they go from haploid to diploid. And so a diploid gamete can combine with a haploid gamete and produce a triploid. Or a diploid gamete can combine with another mistake, can diploid gamete to produce a tetraploid. And so in groups where this is thought to be, where it's thought to exist, you can get these kind of polyploid series. The most common way is thought to be this triploid bridge, and where a unreduced gamete produces a triploid, and then unreduced gamete of the triploid combines with a reduced haploid gamete to produce the tetraploid. Now, in Vaccinium, that's probably not happening, because there's a known strong triploid block in Vaccinium. Probably the way that it's happening is that there are these two unreduced gametes that combine, which is not going to be as common. But the other possibilities aren't really likely either, because one involves selfing. The hybrid bridge involves selfing, and that's where a hybrid actually produces two unreduced gametes, and then they combine through selfing. But Vaccinium saniococcus species are obligate outcrossers, so that's not very likely. And it's also been thought that somatic doubling itself, where a mistake in the mitotic cycle produces a tetraploid, and then that finds its way to the germline and produces fertile tetraploids, that's probably not so likely either. So Camp developed this model for the true blueberries and its evolution. He was one of the first to use ploidy data in taxonomy and propose a model for how polyploidy creates these different lines and species. He hypothesized 25 species, and basically he considered any ploidy difference a species. This is his model. So the horizontal arrows mean autopolyploidy, and the angled arrows mean a combination of allopolyploidy and through those parental species indicated. And so some evolved through autopolyploidy and others through allopolyploidy. And then went from tetraploidy to hexaploidy sometimes, and then sometimes he hypothesized a very complex situation, and not entirely clear how he came up with some of these ideas, but there we have it. At least we have a model to test. He also distinguished between a general high bush type and a general low bush type, 1 meter tall, less than 1 meter tall, or more, or less than 1 meter tall. And he did a lot of crossing experiments and found that species that are at the same ploidy level can cross freely. Those of different ploidy level can't cross. They just don't want to cross. They don't want to produce viable hybrids. And so this influenced his thinking, also thinking that hybridization is rampant in nature, actually. He even hypothesized a species. He named a species, and well, it already had a name. But the species Vaccinium arcansanum was a species, a theoretical species to him, because he thought it was going to be a tetraploid based on a diploid that he saw, and was expecting the tetraploid to actually be found at one point or another. Another researcher later on referred to this as less a taxonomic category than a philosophical concept. It's kind of hard to disagree with that. So this is kind of what we're facing as this first model. Then Sam Vanderklut came along, and he was a professor at Acadia University in Canada, And he devoted his entire career to Vaccinieae and its relatives. A huge body of work, many, many publications, just excellent work for the most part. But I'll talk to you about some differences of opinion I have with Cloet's work. And so he was a proponent of phonetics, and phonetics at the time was hot stuff. It's fallen out of favor now quite a bit. It's overall similarity relationships, and it's not just favored very, very well anymore. But all of his taxonomic revisions had to include this phonetic analysis. He also used elements of biosystematics for taxonomic conclusions, like the same kind of crossing studies that Camp made, seed viability, pollen viability, et cetera, and that also kind of influenced his thinking. So comparing Camp to van der Cloet, van der Cloet basically lumped a bunch of species that Camp had recognized. The reason for that is because van der Cloet believed that cytotype differences don't translate necessarily into different species. And so he lumped things into Angustifolium and Pallidum and a huge pile of things, all high bush blueberries, into this Vaccinium corymbosum in the very broad sense of the word, of the term. And he called corymbosum then a compilo species. Well, what is a compilo species? Compilo species was thought to be a genetically aggressive species which acquires the heredities of closely related species through hybridization and introgression. Sounds like a very nasty, nasty species. I wouldn't want to be around that species. But this is controversial, and the whole concept of compilo species is controversial. It's not well tested. And so nonetheless, since van der Cloet was the expert in Vaccinium and he did the Flora North America treatment, that's the standard. Currently, van der Cloet is the standard for the taxonomy of the true blueberries, and actually all Vaccinium in North America. All right, so there have been some treatments post van der Cloet. They've all been regional, however. They're important, but they have their limitations by area. So there was one in Florida, one for Virginia, and Alan Weekly has done one for the Southeast U.S. based on, I think, primarily on those works. And the general consensus of those treatments is that, well, they maintain the high bush versus low bush concept. 1 meter high, less than 1 meter. And Camp divided the group too finely, they think. They combined several ploidy levels into various species, but also they think that van der Cloet divided the group too broadly. So they recognized several segregates of B. corymbosum. And they considered natural hybridization much less rampant than either van der Cloet or Camp, based on what they're seeing in the field. They could pick out these things in the field, they thought, but they weren't confident about this. Because there were regional treatments, and other folks dealing with regional floras also see some of these entities, or they think they do. But there again, Flora North America treatment is there. Well, something else must be going on where they're mistaken. Well, I'll get to some of that later. So this as a context, I and my colleagues, we wrote a grant proposal to National Science Foundation, and we were funded. It's called an investigation of the evolution and taxonomy of the true blueberries, and it's through the Systematics and Biodiversity Science Cluster. And my colleagues are Paul Manos at Duke University, Hamid Ashrafi and Andrew Kroll at North Carolina State University. And it's a 3-year grant. It started in December. We're just kind of getting our feet wet now. And of course, with COVID, there's been some delays. But I think we can start to do, Andy's already done some field work, and I'm going to be hopefully doing some field work in May. Andy is the postdoc. And we are going to have 2 master's students on the project, one from NCSU, one at Texas Christian University again, joint co-advisor with John Horner, who's the official advisor of record. But we have BRIT and TCU have a MOU where we're at graduate faculty adjunct status there. And one of the products is interesting, going to be a children's book on blueberries. And this is going to be from the BRIT Press. And we have, I think, two qualifications for writing this children's book. Number one, we know something about blueberries, and number two, we all have children. So I'm not sure if those are the greatest qualifications for a children's book, but it's a start. I think we're going to need a ghostwriter. So the goals of the project basically are to look at polyploidy in this group over geography and for character variation within the true blueberries, and produce the first diploid phylogenomic hypothesis of the clade, and then use that and look at how polyploids have evolved over that tree, and look at the possibility of cryptic species diversity, for example. And finally, we're going to revise the taxonomy of the clade through synthesis of morphology, common garden experiments, field data, and the data from the genomics. Okay, briefly the methods. We'll do some field work, a lot of field work, to sample as much as we can throughout the ranges of these species. And we'll take herbarium vouchers, leaf material, photographs, notes on plant, field characters, habitat, and all the vacciniums that grow with our collections. We'll conduct flow cytometry. That's a modern way to estimate the ploidy levels of species of plants, and we've already generated a couple of hundred of those data points. We're going to do phylogenomics then to estimate taxon origins and relationships. And then we'll do a large herbarium study, studying thousands of herbarium specimens, doing databasing, and then producing this monograph as a major product. So a little bit about the phylogenomics, not too much detail here, although it can be very complicated. We'll use the angiosperm 353 probe set with paired end Illumina sequencing and the Hyde-Piper pipeline that's becoming quite popular now. And we'll generate also a whole plastome data set. We'll conduct allele phasing. That's basically allele separation. That's going to be important for differentiating the polyploids. And we'll also generate a diploid nuclear and plastome phylogeny. So we'll use various statistical methods to tease out autopolyploidy from allopolyploidy. I don't really have time to go over those methods, and they're kind of, some of them are admittedly in the experimental stage and need to be refined. So we'll be working a lot on that. But the basic idea is that how do you tell kind of phylogenetically an autopolyploid versus an allopolyploid? The basic idea is that it's like, for the case of autopolyploidy shown here, you can have a single origin, like here, right there, for a polyploid from within a species. You can also have it evolving several times here from a species, or even multiple times, even within populations. It might keep throwing off these cytotypes, for example. For allopolyploidy, the kind of basic pattern you might expect is that the allopolyploids will group respectively with their parents. And so species X here, species Y down there, and then they group with each parent. Each genome of that polyploid subgenome groups with one parent or the other. And again, you can have that occurring a single time, or you might even have that occurring multiple times. But anyway, these are the kind of basic patterns we would expect and hope we can use to test allo versus polyploidy with. So that's all I'll say about that. The rest of this will be mainly about the herbarium study that I and my colleagues conducted. I basically led this part of the study, and it's one of the reasons is because we have a vast amount of material of true blueberries in our combined BRIT herbarium. That's a combination of Southern Methodist University Vanderbilt herbarium and herbarium recently acquired from the University of Louisiana at Monroe. And that is about 3,000 specimens at least. And so a rich source of material for studying these blueberries. You'd think with herbarium specimens, maybe they've already been looked at and annotated and everything is known. Far from the case. I think every time I look at a herbarium specimen, oh, there's a slight exaggeration, but you can always find new things if you look. If you look 10 times at a specimen, the 11th time you're going to see something different. And that's exactly what happened here. And so on the basis of these and other observations, I've been able to come up with this. I haven't talked too much with my colleagues about this yet, but just an idea that can be shot down, or the data can shoot it down. That's totally fine with me. At least we've got something to work with for the true blueberries to test with molecular data. Actually, this I should clarify, that new hypothesis refers to just one little part of the true blueberries. We've come up with other hypotheses that we're testing, with our study. Okay, so first the low bush blueberries. I put low bush in quotes because you'll hopefully convince you that it's an artificial category. And I'll put the putidiploidy in parentheses when we go over these. First, there are kind of like non-controversial species. There's like the species Vaccinium hirsutum, for example. That is a narrow endemic to the southern Appalachians, and it's easily distinguished from any other species by those weird glandular hairs all over the calyx. Those hairs are even on the corolla lobes, and it's very distinctive. Then there's Vaccinium tenellum, and a pretty easy one to identify. It's deciduous, and it's got these stipitate glands all over the backs of the leaves, and those glands have a really kind of elongate head on them. The head is like this whole thing taking up much of the length of that stipe, or the what looks like a stipe. And then there's two others. These are widespread northern species. And one is angustifolium, the other is myrtleoides, distinguished by cerulate leaves versus entire margin leaves. And there's a few other characters, and not too much going on there. There's possibly a diploid version that's been recognized as boreali by van der Kloet. Okay, then there's Darrowi and Mercenites. And Darrowi and Mercenites, I think they're one and the same thing. They're evergreen low blueberries that occur in the coastal plain and in sandy soils and pine forests and things like that. And they're these evergreen plants that are quite distinctive from the rest of the group. But sometimes they're glaucous, sometimes they have blue fruit, sometimes they have black fruit. Sometimes they have these stipitate glands on the leaf backs, and sometimes they don't. And it's thought to be correlated with ploidy. I'm not convinced that's at all correlated with ploidy, and so that's maybe something that the master's student will be investigating to see if there are these correlations. Then there's Vaccinium pallidum, the hillside blueberry, and this is the most widespread probably blueberry and common blueberry you might encounter if you're in the field. And so this is distinguished by their pallid, they're kind of glaucous looking, they have relatively small flowers. The problem is a lot of times it's hard to distinguish this from some high bush blueberries on herbarium specimens, because herbarium folks doing these collections do not, or they fail to provide, as E.G. Voss is mentioning here in his treatment, they often neglect to note the stature of the plant. And that's really unfortunate, because it would be much easier to separate these by stature in this particular case. Ed Voss knew what he was talking about. And so please, if you're collecting, always record the height of the plant. It doesn't get reported on, you can't see how tall a plant is on herbarium specimens unless you're picking the whole plant. So that's very important. Also, record the other features that you think won't record well upon drying. And so then there are these plants, for example, in Arkansas, that are taller than they're supposed to be. Plants somehow don't follow the rules all the time. They are supposed to be less than 1 meter, and these are 6 feet tall. And so otherwise they look like pallidum. So what are they? That's one question we need to ask. The folks in Arkansas, the field botanists I know, they see these plants too, and they're wondering the same thing. Then there are these Appalachian things. Appalachians are probably the most complex area of blueberries, true blueberries, where things can go a little haywire. You can get things that are tetraploid, things that are hexaploid, different looking tetraploids, and maybe they're derivatives of pallidum, maybe they're crosses. We just don't know. There are hypotheses out there, all kind of open for testing with our data. Then there are the high bush blueberries. Okay, the first one is, to me, this is Elliottii, clearly distinct from corymbosum. It's fairly easily recognized by not only its small leaves, but also the fact that, and this was what I found during a herbarium study, is that the styles in the flowers are included. And the prior experts thought that there wasn't that much character variation in the flowers and they weren't very useful. This is really practically diagnostic. You can compare like the style of virgatum, for example, and how highly exerted it is out of the corolla. And it's very straightforward. So here is a van der Kloot's thought. Van der Kloot has passed away now. I'd love to have talked about him about this, but he even wrote a paper, Vaccinium Elliottii, once more with feeling. Oh my gosh. Okay, so of all the taxonomic decisions I have made, retaining Vaccinium Elliottii in the corymbosum sensuletto complex has been the most controversial. Nonetheless, asking those who object most vociferously to this transfer to state what characters they would choose to separate Elliottii from corymbosum, they inevitably refer to the plant's gestalt. Okay, well, and then he goes on to say later in the paper, while attempting these reciprocal crosses, I discovered that all Elliottii flowers had styles that scarcely exceeded or were level with the length of the stamens, regardless of the length of the corolla tube. And I'm reading this and I'm going, and you still lumped Elliottii with the rest of the corymbosum complex. Why? So I'm mystified by this, but he was very stubborn apparently this way and had his kind of concept about corymbosum, and that was going to be his concept, despite other evidence to the seemingly contrary, in my opinion. Then there's Vaccinium virgatum, and this one is also quite distinctive to separate from corymbosum. It's got these kind of rhomboid leaves, generally speaking, but the telltale sign is these little glands on the backs of the leaves. They're different looking than the ones in the Myrsinites Darrowiii group, because the heads are very rounded. They're not these elongate things, so it's really easy to tell them from the other ones. And Camp recognized a tetraploid and a hexaploid in this group. Morphologically, I can't detect any difference in ploidy based on like plant size or parts size or anything like that. So that has yet to be tested. Okay, so after that, then there's this residual high bush species. So it's all the species that are left over after those are pulled out. And these basically have, it seems, highly inconsistent differences in leaf size, leaf margin. They can be serrate, they can be entire. There's pubescent size differences, an arrangement, flower size, fruit color, and it's thought that the tetraploid glabrous version of this is actually considered to be the progenitor for most of the high bush cultivars that we have in cultivation now. It's referred to now as Vaccinium formosum. It used to be called Australia, but that's just a nomenclature issue. And so this kind of has relevance for the origin of our cultivated blueberries. What is this origin? What is this thing that gave rise to these cultivated blueberries? Well, there's a great paper that was out a couple of years ago showing that, in fact, diploids and tetraploids can occur in the same population. On the right is a tetraploid plant, and on the left is a diploid plant of what would be known as Vaccinium fuscatum or corymbosum in the wide sense. And they're entire leaf plants that are pubescent. And interestingly, this is probably the fulfillment of Camp's prophecy of the tetraploid arcansanum that he was referring to in a philosophical sense. I think this would make Camp very happy to see this. And most interestingly, in this population, they found an actual flowering time difference of a week. The tetraploids flower a week later than the diploids do. And so there's probably some isolating mechanism going on here. Another interesting kind of fact about this group is in Uttall's Vaccinium treatment of Virginia. So at the 4N level, he recognized 3 different species, one of which is completely glabrous, that would be Formosum, one of which is densely pubescent, that's Fuscatum, and then he has this Marianum, which is basically sparsely pubescent, and some maybe other intermediate features. And notice the distributions of these things are basically all the same in Virginia. And that really made me wonder, are these really distinct species, or is this kind of just variation at this ploidy level? And then I found this specimen from Michigan. This is a really interesting specimen because it kind of confirms a suspicion of these things kind of occurring together, but really staying separate. And in Michigan, they're all lumped into Corymbosum. But this specimen has 2 branchlets on it. And the one branchlet has cereulate leaf margins, and it's in flower, and it's pubescent. The other branchlet, well, the leaves are entire, and they're glabrous, and it's already well on to its way to mature fruit. And so it looks to me like these 2 specimens are doing completely different things, but growing together. And I suspect that the thing on the right is the same thing that of the tetraploid they might have found in that study, and that the cereulate thing is doing its own thing, and that would be maybe Corymbosum sensu strictu. Anyway, I put this all together and have this kind of residual high bush blueberry hypothesis. So what maybe is going on, and this could be refuted tomorrow, and based on data, is that we've got a 2N entire early flowering thing, which is Fuscatum, and that can be glabrous to pubescent, that's trivial characters. Then we've got an autoploid 4N entire late flowering entity, and that would be Arcansanum, so-called. And that's also glabrous to pubescent. There are these might be rare or single events based on this kind of idea of a triploid block. Okay. Then you've got a 4N cereulate thing, which is Corymbosum sensu strictu, which occurs widely across the northern parts of the range. The origin would be uncertain. Maybe it's there's an extinct diploid, or maybe there's a diploid hanging around that's yet to be sampled. So what this would mean was that it would be that cultivated high bush blueberry is really derived from, it's not derived from a wild species per se, but just a selection within this kind of glabrous version of this Arcansanum, and it's been back crossed to produce a stable glabrous glaucous fruited line. And Vanderkloot did a little study and anecdotally noted that in progeny from parents that he grew up, that pubescence quantity and fruit color were not really true breeding in the wild. And so this might affect this hypothesis as well, or support the hypothesis. The problem is he didn't really document the study with images, didn't make it a separate study, and didn't make vouchers. So it's really, we can only treat it as anecdotal at this point. Okay, so just a little bit of molecular data. Just some initial results that Andy and Paul have now. And these are, the exciting thing about this diploid, we did the diploid phylogeny. The most exciting thing for me about this diploid phylogeny is that the Caesariense fuscatum, that's that residual high bush complex up at the top there, is separate from pallidum. And so that makes me very comforted to think that we can actually get resolution between pallidum and the rest of this high bush complex, and hopefully it'll be stable that way. And it's really just on herbarium specimens, that's just the fact that we can't tell one from another sometimes is just an artifact of folks not putting the height down on the specimens. Okay, then we've got the diploid phylogeny. Now we've put polyploids into the analysis, and for those in the know, this is a consensus. This is not allelic data. This is just a consensus sequence, and it's all 300 or so genes have been concatenated together, and there's just a maximum likelihood analysis run on those. But nevertheless, there's still some powerful results here, I feel, because things are quite stable relative to the diploids and the polyploids. The residual is all up here. They're all grouping together with nice support. And all the glandular ones are grouping here. That's not glandular, but it's these are glandular. And then pallidum continues to be separated from that clade. And we've got this kind of Southern Appalachian, Northern North America group here, which includes this oddball 6X, whatever it is, simulatum, and 4X altomontanums. So there's a lot more that needs to be done with this clade, but we're getting nice resolution. And we have a plant that was a known hybrid allotetraploid that's grouping in the right place here. This is Myrtleoides times Eliadii, and it's grouping strongly with Eliadii as kind of a control. All right, so with that, I'd just like to say a few words about specimen data mobilization, because this is part of a WeDig Bio event, and I've actually used digitized data quite a bit, even in preliminary data, generating herbarium observations and planning out field work with digitized data. So what is data mobilization? Data mobilization is the digitization of the specimen. So that is the image, label transcription, other transcription from some things associated with the specimen, and georeferencing, estimating where it was collected. Because sometimes the label information is quite vague, unfortunately, and a lot of them were collected before GPS. And then dissemination through online portals. Okay, so we have several grants running at the same time right now at BRIT, and all in collaboration with other organizations. We have one called TORCH, Texas and Oklahoma Regional Consortium of Herbaria, and that's led by us, and that's digitizing all specimens collected in the states of Texas and Oklahoma in 40 plus herbaria, many throughout Texas and Oklahoma, and then several scattered around major herbaria such as Missouri Botanical Garden, New York Botanical Garden, et cetera, that have historical collections from those states. We have one called Endless Forms, Most Beautiful and Wonderful. Those are specimens with digitizing plants that are, say, succulent plants or unusual growth forms or nutrient uptake, such as parasites and other growth forms like epiphytes. And that's led by New York Botanical Garden. And that includes neotropical vacciniums. And so all the epiphytes are going to be, all the Vaccinieae are going to be databased in the neotropics because of this grant. Then we have the fern grant, and that's led by UC Berkeley, all our ferns digitized. And then we most recently was able to recruit funding from Google Arts and Culture. So this preserves and brings the world's art and culture online. Well, to the extent that this is art, it can be considered art, I guess, and also culture. So they're digitizing our neotropical trees. And we've just hired a digitization tech, Jarek Brown, I believe his name is, for digitizing these specimens. And BRIT collections will be featured in the upcoming GA&C International Project trees. We've got other completed and pending grants going. Some benefits, this enables virtual access for those unable to visit collections in person. It reduces travel costs, wear and tear on the specimens. We can do a lot of record keeping. We can do a lot of queries that were not previously possible. And specimens can be aggregated across collections for big data analyses, and it preserves a digital record of specimens that might be subsequently lost or damaged. And I want to focus on that last point, because here is a really damaged specimen of Vaccinium. This was from northern Louisiana, Monroe, one of the herbaria that we acquired recently, and they had bad bug damage. We got rid of all the bugs. There's no bugs in that collection anymore, but the damage had been done. Cigarette beetles can rifle through collections pretty quickly, and you can see that there are really no flowers left on this specimen. And that's really unfortunate because there are no leaves out yet on this particular plant, the flowers before the leaves. And so we don't have much data here, and I can't really identify it. However, several years prior, the specimen was imaged. And so in this one, this is the same specimen when all the flowers were intact. And so now it's easy to identify because we've got these open corollas there at the top, and they don't have styles exerted. It's Eliadii. So this is kind of a really interesting way that digitized specimens can be utilized. So another benefit would be for selecting sampling localities. I've used these resources a lot recently to try to figure out where to collect vacciniums. And so there are several ways you can do that. You can walk around and try to find them. That's probably not productive. You can go word of mouth and find out where other people have found them. Probably more productive is to use physical specimen data, online specimen data portals now. And there's a relatively recent innovation called iNaturalist. And so comparing those, well, I should say a little bit about iNaturalist. iNaturalist, people go around and take photos, and that photo is automatically linked to a geo-coordinate, so you know exactly where they took it. And people post on the site, and it's becoming extremely popular. So specimen portals versus physical specimens, advantages, disadvantages. The big advantage is the large expansion, obviously, of locality options. Unless you've got many loans, you can't possibly get all that many loans in. So this data is now at your fingertips. Having said that, there's still some challenges involved, so that the data are still very, very gappy. It's very frustrating to only have, say, skeletal data, only, for example, up to county. They don't say exactly where it was collected. That data is not entered yet. They're often lacking images still. And the image quality can vary, and the resolution varies quite a bit too. And so resolution is going to be very important for those micro characters, like the glands, for example, on the backs of the leaves. Sometimes the packets aren't opened. And sometimes the accuracy and precision of the coordinates varies all over the board. That's true for labeled data, but it's also true for estimated geocoordinates where, you know, the precision maybe is not as good. So you have to go in, and sometimes you can figure it out maybe a little better. So then what about iNaturalist versus specimens? And I'm sorry this talk is going over, but I'll be finished in a few minutes. There is iNaturalist versus specimens. The photo is usually directly linked to geocoordinates, as I mentioned, and it's relatively recently documented. So you can be fairly confident you can go there and still find the plants. That locality hasn't been this, you know, it's not a shopping mall now, for example. And then you can have precision of the geocoordinates varying quite a bit in iNaturalist. So often they're even intentionally masked, so that's a drawback. Photographic quality, of course, can vary. Photographic resolution can vary. The photographs have appropriate features. Sometimes they'll take 10 shots and it's not the shot that you want for the character that you're looking for. And sometimes they're in private settings like backyards that you probably can't get easy access to. There's uncertainty as to whether the plant is cultivated or not. Sometimes I see in the background rows of blueberry plants, and I see, oh, okay, that's probably from a blueberry farm, which I don't want. And then you have an indication, you have no indication as to how to get to the locality sometimes. And then field notes are not usually taken on iNet photos. And then iNet search functions are actually limited, which I think hopefully they will be improving at some point. And with that, I kind of will just wrap it up. And this being kind of a We Dig Bio event, I do want to mention that the event continues. It's already we already had an event yesterday, and on the 10th and 11th we'll have a virtual transcription blitzes from 9 to 10 a.m. Central Daylight Time, and then virtual live tours and presentations from 10 to 11 a.m. Central Daylight Time. For more information, visit www.brit.org/WeDigBio, and it would be great to see you virtually there at these events. And with that, thanks very much for attending, and I will take any questions that you may have thought of. Thank you.
Thanks, Peter. That was fantastic. You did a really good job. Okay, so we have a number of questions. Some are still coming in too, since we've got a little bit of lag from here over to YouTube. But so this goes back a little bit in your talk. So there's a question from Harold Keller. You can see it here on the screen. He says, hello, how important are chromosome numbers, ploidy levels, and karyotypes today? And has genomics replaced these data formerly used to determine plant relationships?
Well, that's a good question, Harold. They're extremely important, but maybe for a different reason than before. Before, they were using things like karyotypes, morphologically. They would look at the appearance of the chromosomes and kind of make conclusions of relationship based on that. That's not done so much anymore. That's been replaced by DNA sequence data to a large extent. But they're just as important because you really, with a plant group that's undergoing ploidy of any sort, it's really hard to get a grasp of their details of their evolution without knowing that ploidy number. And with these new tools, flow cytometry in particular, it's just so much easier now to sample ploidy level than it ever was before. Before we had to do the old thumb squash technique, and it took a lot of time, and people just kind of didn't want to do that kind of work. And because the amount of work that it took in order to get point data was just so there wasn't a lot of return for what you got. But with flow now cytometry, it's a game changer. It really is.
Excellent. Okay, we've got a couple questions from Morgan Gustell, who says, I may have missed the beginning, but he has 3 quick questions. So here's the first. What are the estimated genome sizes, and thus how feasible might be whole genome sequencing? I believe somebody else tried to answer that, but you probably know offhand.
Yeah, should we go over the other questions? I'll just answer that. He's got a couple other questions there. To the extent of knowledge, it's not so large that you can't do whole genome sequencing. In fact, I didn't mention this, but we are going to do some whole genome sequencing on that residual high bush group, because it turns out, and we put this in the grant proposal because we already sort of suspected that this was going to happen, but we didn't get enough resolution in that residual high bush clade, even with, they're telling me, hard to believe, 300 loci. But that's the way it appears. And so we're going to actually do whole genome sequencing. We've got a reference genome already with Vaccinieae corymbosum that was done very recently. And so we'll be able to use that. And obviously it's more expensive to do that. And so we'll only do that for a subset of the samples. At some point, my understanding is that maybe it would switch over to entirely whole genome sequencing, but I don't think we're quite there yet.
Okay, I think you kind of answered all his questions naturally. Okay, let's see. Do we have another one over here? Sorry, I'm juggling these multiple screens. Thank you again, everybody watching, for being patient and following us over to the second link. It went off. Peter, you maybe weren't aware, but it went smoothly. Nothing went wrong. Nothing went wrong.
Perfect.
Let me see. It looks like, yeah, someone tried to answer. Morgan said, and Andy Crowell said, the diploid Vaccinium genomes are in the ballpark of 600 MB. So
thank you, Andy.
Yeah. They're having a little chat here in the talk. Just a lot of thanks, a lot of praise. I mean, really, it was super thorough, so I'm not surprised that there aren't a lot of unanswered questions. I do have one. Let me see, double check over here. Yeah. I have to ask you just personally, so what would be saying that you work on blueberries is a great way to kind of capture the imagination and the attention of the general public. But if there's something, one kind of singular thing that you would say either is best about them or that you can tie into some kind of larger message, what would it be? I know that's tough. Sorry about that.
It's clearly it has, you know, it's not an applied study per se, but it's a basic study that has the potential for the data to be used for all sorts of applied uses. Obviously, in blueberry breeding, to make a better blueberry, and there's really not. I don't think there's a ton of research. There's got to be resources out there, but this will just add a huge. There's resources, but a lot of them are based on plants that are in the greenhouse already or are already cultivars, not really from natural sources like we're using. And so it's really going back to the basic stock and asking what is the genetic variation like in that natural stock, that could be used for blueberry breeding in the future.
Yeah, I couldn't help but thinking as soon as you showed those like glandular hairy ones going, but can you eat them? And what would that feel like? Would it be like a peach versus a nectarine?
Yeah.
Who knows? All right. Well, thank you so much
Right.
for giving us your time today. And watchers, if you're still with us, don't forget to check us out next month on May 4th for another talk from the researchers from the Perot Museum. We'll see you next time. Thank you.